Welcome back, fellow time-traveling geneticists, to Decoding Dynasty. If you joined us for our last episode, you'll know we took a deep dive into the life of Emperor Ferdinand I of Austria, unraveling the genetic and medical mysteries behind his struggles with epilepsy. Well, Ferdinand's story is so packed with fascinating scientific puzzles, we just had to give him a second episode. Today, we're continuing our journey into his unique medical history, turning our genetic magnifying glass to another significant challenge he faced: hydrocephalus. Often referred to as water on the brain, hydrocephalus is far more complex than that simple phrase suggests. It's a condition that can profoundly impact development, brain function, and overall quality of life. And for Ferdinand, who lived in a time when understanding of the brain was still in its infancy, this condition undoubtedly added another layer of extraordinary difficulty to his reign. So buckle up. We're about to explore the delicate balance of cerebrospinal fluid, the genetic factors that can disrupt it, and how this intricate condition likely affected the mind and life of Emperor Ferdinand I. Get ready to decode another crucial piece of the Habsburg puzzle. Just like epilepsy, the story of hydrocephalus often has roots in our genes, although the specific players and pathways can be quite different. The genetic landscape of hydrocephalus is complex, to say the least. Scientists have linked over 100 different genes to this condition, highlighting just how many different things can go wrong in our genetic code to disrupt the delicate balance of fluid in the brain. For most folks with hydrocephalus, it's not down to a single, easily identifiable gene mutation following a simple inheritance pattern. Instead, it often involves a combination of subtle genetic variations, possibly interacting with environmental factors, a complex genetic cocktail, if you will. However, there are some specific genes where a single mutation can have a more direct and significant impact, particularly in cases of congenital hydrocephalus, meaning it's present at or around birth. Let's look at some of the key genetic culprits. One important gene is FOXJ1, located on chromosome 17q21.33. This gene is crucial for the development and function of those tiny hair-like structures called cilia that line the ventricles of our brain. These cilia act like little oars, helping to move the cerebrospinal fluid (CSF) around. Mutations in FOXJ1 can mess up how these cilia form and work, leading to both communicating and obstructive hydrocephalus. Basically, the fluid either can't flow properly or can't get where it needs to go. We can see different inheritance patterns with FOXJ1 mutations depending on the specific mutation. Another gene, HYDIN, though primarily studied in mice, has shown that frameshift mutations, imagine a typo in the genetic code that shifts the reading frame, can cause severe communicating hydrocephalus by affecting the ciliated ependymal cells, which are the cells lining the brain's ventricles and sporting those all-important cilia. The human equivalent and its precise inheritance pattern are still being fully unraveled. Then there's CCDC39, found on chromosome 15q6.1. Mutations in this gene also result in abnormal development of those motile cilia, leading to impaired CSF flow and, in animal models, neonatal hydrocephalus. Again, this highlights the critical role of cilia in maintaining the brain's fluid balance. Interestingly, researchers have also found damaging mutations in AK9 and several other genes that are highly active in the ventricular lining in patients with idiopathic normal pressure hydrocephalus, INPH, a form that typically affects older adults. These mutations in genes like AK9 located on chromosome 7p15.2 seem to affect the beat frequency of cilia and the overall dynamics of CSF. Moving beyond cilia, mutations in the MPDZ gene located on chromosome 7q11.23 can cause multiple malformations in the ependymal lining itself, disrupting the tight junctions between these cells that are crucial for maintaining the integrity of the ventricular system. This can lead to congenital hydrocephalus. A gene called TRIM71, found on chromosome 8q24.3, has been linked to a neurodevelopmental syndrome that includes ventriculomegaly, enlarged ventricles, and hydrocephalus. De novo mutations, those that pop up spontaneously and aren't inherited, in TRIM71 seem to affect the function of neural stem cells, which are the building blocks of our brain. Finally, mutations in B3GLCT, located on chromosome 11q25, which is associated with Peters plus syndrome, can also lead to hydrocephalus. This gene affects both cilia and the secretion of proteins in ependymal cells and a structure called the subcommissural organ, further underscoring the complex interplay of factors involved. It's important to remember that for many of these genes, the exact inheritance pattern can vary depending on the specific mutation. Some might be autosomal dominant, others recessive, and some might even be X-linked. What this all tells us is that the genetic roots of hydrocephalus are diverse and often involve disruptions in the development and function of the brain's fluid management system at a very fundamental level. Now let's zoom in even further and explore what's happening at the cellular and molecular level in the developing brain when hydrocephalus occurs. Understanding these tiny processes can give us big insights into how this condition arises. Think back to how a normal brain develops in a fetus. It's a beautifully choreographed dance of cells dividing, proliferation, and becoming specialized differentiation. Neural stem and progenitor cells, NSPCs, are the stars of this show. They multiply in a regulated way to create all the different types of brain cells we need: neurons, astrocytes, support cells, and those crucial ependymal cells that line the ventricles and sport the CSF-moving cilia. This coordinated growth ensures the brain forms its organized layers and that the ependymal lining is fully functional, helping with CSF flow and overall brain balance. Proteins like VPS35 are even involved in making sure these ependymal cells develop and survive properly. But in hydrocephalus, this cellular symphony goes a bit off-key. We often see a reduced proliferation of those neuronal precursor cells, meaning fewer neurons and glial cells are produced. This can be triggered by genetic mutations or even environmental factors. At the same time, the differentiation of NSPCs into ependymal cells can be impaired. This leads to fewer ependymal cells, and the ones that do form might be dysfunctional, with abnormal or even missing cilia, directly impacting CSF movement. Interestingly, hydrocephalus is also linked to increased cell death, or apoptosis, near the ventricles. This loss of essential cells, coupled with abnormal proliferation in some areas, further disrupts the delicate cellular architecture of the developing brain. And if the connections between these developing cells, the cell junctions, are also abnormal, the structural integrity of the ventricular lining can be compromised, making the hydrocephalus worse. Now, let's delve into some specific molecular players. We talked about genes earlier, but how do their mutations actually cause these cellular problems? Well, mutations in key regulatory genes like TRIM71, L1CAM, MPDZ, CCDC88C, and NFIA can really throw a wrench in the normal proliferation and differentiation of neural stem and neuroepithelial cells. This can lead to premature specialization of these cells, meaning fewer are left to become neurons and glial cells, resulting in an abnormally formed brain, sometimes with a more compliant, less rigid cortex. This altered brain structure can then contribute to the ventricles enlarging, even if the initial CSF flow isn't directly blocked. This idea shifts some of the focus from just CSF flow problems to these early brain development defects. Think about those cilia again. Their movement is crucial for CSF flow, and the proper functioning of their internal scaffolding, the microtubules, is essential. Mutations in genes like CATNAL2 can disrupt these microtubule dynamics in fetal radial glia, a type of neural stem cell, and their descendants, leading to wonky cilia and ultimately ventriculomegaly and hydrocephalus. Even signaling pathways, those communication networks within cells, can be affected. For example, specific mutations in TRIM71 can alter the WNT beta-catenin signaling pathway, speeding up neural differentiation in a way that contributes to hydrocephalus through unique mechanisms. Finally, let's consider the role of the brain's immune cells, the microglia, and those vital ependymal cells. When hydrocephalus develops, microglia can become activated, leading to neuroinflammation. They release inflammatory molecules that can cause further damage to neurons and even contribute to behavioral problems. On the flip side, the survival of ependymal cells is critical. They maintain the barrier between brain tissue and CSF and help the CSF flow smoothly. If these cells die or detach, a process called ependymal denudation, that barrier breaks down, and CSF can accumulate. In some cases, especially in the narrow passages like the cerebral aqueduct, the loss of ependymal cells can even lead to tissue fusing together and causing an obstruction. Interestingly, there are repair mechanisms where other cells try to fix this damage, but if the damage is too severe or the repair is too slow, the hydrocephalus can progress. So, at the smallest level, hydrocephalus is a complex interplay of genetic glitches leading to disrupted cell proliferation and differentiation, faulty cilia, altered signaling pathways, neuroinflammation, and the loss of those crucial ependymal cells that manage our brain's fluid. Now that we've peeked at the genetic blueprints that can sometimes lead to hydrocephalus, let's talk about what this condition actually looks like and what's going on inside the brain. Hydrocephalus, at its core, is all about an abnormal buildup of that vital brain juice, cerebrospinal fluid, or CSF, in the brain's ventricles, those interconnected spaces within our brain. This excess fluid causes these ventricles to enlarge, and that increased volume can put pressure on the surrounding brain tissue, leading to a range of neurological symptoms. The way hydrocephalus shows up can be quite different depending on when it develops. Whether it's congenital, meaning present at or around birth, or acquired, developing later in life due to things like infections, bleeding, head injuries, or inflammation. In infants and young children, one of the most striking signs is a progressive head enlargement, or macrocephaly. Their heads can grow at an unusually rapid pace. You might also notice a tense or bulging anterior fontanelle, that soft spot on the top of a baby's head, and the seams between the skull bones, called cranial sutures, might be visibly spread apart. Sometimes the veins on the scalp can also appear more prominent or distended. These little ones might also experience frequent vomiting, be unusually irritable or drowsy, and show delays in their developmental milestones. Interestingly, it's been noted that up to half of infants with hydrocephalus might not show any obvious symptoms initially, even if the pressure inside their head is rising. In older children and adults, the symptoms tend to be more related to that increased intracranial pressure. A common early sign is a persistent headache. They might also develop problems with their gait, struggling with walking, appearing unsteady or having a wide-based stance, and experiencing balance issues. This is particularly noticeable in a specific type called normal pressure hydrocephalus, NPH, which often affects older adults. Cognitive impairment is another significant issue, with people experiencing memory loss, difficulty concentrating, and a general slowing down of their thinking. In adults with NPH, urinary incontinence or a loss of bladder control is also a classic symptom. Besides these, other neurological signs can include drowsiness, dizziness, tremors in the arms, weakness, and in some less common cases, even psychiatric symptoms like mania. It's worth noting that in NPH, you often see a specific triad of symptoms: a shuffling or wide-based gait, cognitive dysfunction, and urinary incontinence. However, not everyone with NPH will present with all three of these. Now, let's talk a bit about what's actually going wrong with the fluid dynamics. Hydrocephalus basically boils down to a disruption in the normal homeostasis of CSF. This can happen in a few ways. Either the brain is producing too much CSF, the absorption of CSF is impaired, or there's a blockage in the flow of CSF through those ventricular pathways we mentioned earlier. Newer research is also highlighting the importance of other factors, like how stretchy or compliant the brain tissue is, and how water is transported in and out of the brain. Inflammation also plays a significant role, especially in cases of acquired hydrocephalus that develop after a brain hemorrhage or an infection. These inflammatory responses can actually damage the delicate pathways that CSF uses to flow and be absorbed. Inflammation can also mess with the cells that secrete and clear CSF, further contributing to the problem. So it's not just a plumbing issue. Sometimes it's also an inflammatory response that's gumming up the works. Understanding these symptoms and the underlying mechanisms of how CSF flow is disrupted and pressure builds up is crucial for diagnosing hydrocephalus and figuring out the best way to manage it. Now, fast forward to today's medical world. How do we figure out if someone has hydrocephalus? Modern diagnosis is a lot more sophisticated than just observing symptoms. Doctors use tools like imaging scans, primarily MRI, magnetic resonance imaging, or CT scans, computed tomography, to get detailed pictures of the brain. These scans can clearly show enlarged ventricles and assess the flow of CSF. Neurological exams, developmental assessments, especially in children, and sometimes even specialized tests to measure CSF pressure all play a role. Genetic testing might also be used to identify specific underlying causes, though hydrocephalus itself is often a complex condition with many potential origins. And when it comes to treatment, mercifully, modern medicine offers solutions that were simply unimaginable in Ferdinand's era. The primary treatment for hydrocephalus is typically a shunt system. This is a small, flexible tube surgically placed in the brain to drain the excess CSF to another part of the body, like the abdomen, where it can be safely absorbed. It's like installing a tiny internal drainage system. While it's a lifelong management, shunts can dramatically relieve pressure and prevent further brain damage. In some cases, a procedure called endoscopic third ventriculostomy, ETV, can be performed, which creates a small hole in the brain to allow CSF to bypass an obstruction, effectively creating a new pathway for fluid flow without needing a shunt. So, while Ferdinand I navigated his life with both epilepsy and the profound, unmanaged effects of hydrocephalus, individuals today benefit from sophisticated diagnostic tools and effective treatments that can significantly improve outcomes and quality of life. It truly highlights the incredible strides medical science has made, allowing us to manage conditions that once seemed insurmountable. And there you have it, another layer peeled back on Decoding Dynasty. In these 2 episodes, we've journeyed through the fascinating and often challenging world of neurological disorders, specifically epilepsy and hydrocephalus, using the powerful lens of genetics and modern science. Through our exploration, we've seen how complex these conditions are, often with intricate genetic underpinnings involving a multitude of genes and developmental pathways. In the case of epilepsy, we delved into the role of ion channel mutations and the diverse ways seizures can manifest. With hydrocephalus, we examined the delicate balance of cerebrospinal fluid and how disruptions at the genetic, cellular, and developmental levels can lead to its accumulation. As we reflect on the life of Emperor Ferdinand I of Austria, who history tells us suffered from both of these conditions, our modern understanding offers a new perspective. The close genetic ties within the Habsburg dynasty, exemplified by his parents being double first cousins, likely amplified the risk of inheriting recessive genetic factors that could have contributed to his hydrocephalus and severe epilepsy. While we can't definitively pinpoint specific gene mutations without his genetic information, our scientific knowledge illuminates the potential biological realities behind his documented health struggles. Ferdinand's story serves as a poignant reminder that historical figures were not immune to the biological forces that shape our lives. His reign, often characterized as weak and indecisive, was undoubtedly impacted by the relentless burden of his neurological conditions. By examining his life through the lens of genetics and modern medicine, we gain a deeper appreciation for the personal challenges he faced amidst the backdrop of a powerful empire. Thank you for joining me on this genetic journey through history. If you found these episodes of Decoding Dynasty insightful, please like, subscribe, and share it with others who might be interested in unraveling the past through the fascinating world of genetics. And speaking of the Habsburgs and their intriguing medical histories, would you like us to turn our genetic gaze to another member of this influential dynasty, a figure known for his own unique health challenges? Perhaps the last Spanish Habsburg, Charles II? Let us know in the comments below if you'd be interested in decoding his story next. Stay tuned to The Geneticist to find out what historical mystery we'll unravel next.